Okay, ladies and gentlemen, it's about 8:30 on my watch, so welcome everybody to this plenary session. My name's Clive Page, and it's my pleasure to introduce you to Professor Sir Andrew Pollard, who's the Ashall Professor of Infection and Immunity at the University of Oxford. And Andrew's going to talk to us today about COVID-19 and its legacy. Andrew qualified in medicine before doing a PhD, and he also is involved in a number of government committees and national committees. He chaired the UK's NICE Meningitis Guidelines Development Group, the NICE Topic Expert Group developing quality standards for the management of meningitis and meningococcal septicaemia. And as many of you will know, Andrew's research includes the design, development, and clinical evaluation of vaccines, which he's going to cover this morning, including those for meningococcal disease and enteric fever. He's been using human challenge models of paratyphoid. He runs a surveillance for invasive bacterial diseases and studied the impact of pneumococcal vaccines in children in Nepal, and leads a project on the burden and transmission of typhoid in Nepal, Bangladesh, and Malawi. I think it's fair to say Andrew's a very distinguished academic. He received the Bill Marshall Award of the European Society for Paediatric Infectious Diseases in 2013, the Distinguished Award for Education and Communication in 2015, and the Rosenstein Medal in 2019 awarded by the Swedish Paediatric Society and the Swedish Society of Medicine. He was elected a Fellow of the Academy of Medical Sciences in 2016, and is an NIHR senior investigator. Andrew is also the chief investigator that led the development of the ChAdOx1 COVID-19 vaccine against SARS-CoV-2. I'm sure he'll cover a lot of that this morning. And correctly, Andrew, he was awarded in the 2021 Queen's Birthday Honours, awarded a knighthood for services to public health, particularly during the COVID-19 pandemic. Given your CV, Andrew, it's very good that you've made the time to join us today. You're obviously very busy, and we look forward to hearing this morning from you about the legacy of COVID-19 and particularly around the vaccines that you were so important in helping to develop. So, welcome.
Thank you very much, and thanks to the organisers for inviting me to speak to you today. It's a great pleasure to be here. And in some ways, it seems a bit strange to be talking about vaccines now that everyone in the world is a vaccine expert. It seems almost unnecessary to have to talk to anyone about them. But what I'd like to do to start with is to ask you to consider a situation where you're sitting here in Glasgow and you start to hear of a new disease that's spreading in Europe. And that disease then first arrives here in the UK on the south coast, here in Weymouth on the south coast of England. And there are some cases of this new disease described around that time. And over the next few weeks, the cases appear all over the United Kingdom. And you start to hear where you live of individuals who develop an illness where they have a fever for a few days, they may become very breathless, some develop swollen glands, and then sadly quite a number of people die over the ensuing week or so. And without any information about what this disease is, what's causing it, and a concern about what that might mean for you, there is increasing anxiety amongst people around you, and actually people stay at home because they're very worried about how you get this disease and whether they might catch it. And so the streets are deserted. There's no information at all from the government. In fact, they've also left and gone away because they're so worried about catching the disease. And amongst families, people are worried even if their children become unwell because of the implications of a disease which is apparently killing large numbers of people. Without any knowledge of what's going on, can you imagine the fear that transmission of a new disease could cause? And of course, that was exactly the situation back in the mid-14th century when Neisseria pestis began to spread across Europe. It first arrived in Europe in Crimea and rapidly spread across the continent before arriving here in the UK via a ship arriving in Weymouth. And those first cases were almost certainly individuals infected by fleas that carried Neisseria pestis, and they were on the backs of rodents which were on the ships. But then the bacterial infection spread incredibly rapidly across the population of the UK, with almost certainly person-to-person transmission, resulting in this very rapidly fatal condition, pneumonic plague, which was able to kill people within a matter of days of infection. And that's those individuals that became rapidly breathless. As a result of the arrival of the Black Death, known at the time as the Great Pestilence, half of the population of Europe died. It took another 100 years for the European population to return to the levels that it had been at in 1345. So pandemics like the one we've just had with COVID-19 are rather different from some of those of history. I think also this lack of understanding of the microbial world at the time and seeing whole families, whole villages wiped out by an infection added another layer of anxiety and fear on top of that was already there as a result of the disease which people were experiencing. Lack of information, and science was really not there to provide any information or to help. And that really contrasts with the situation with the COVID-19 pandemic. This is a newspaper headline from the New York Times on the 11th of January 2020. I suspect most people in this room had no idea that there was even an outbreak in Wuhan in China on the 11th of January, and certainly were not expecting a pandemic. But on this day, the Chinese scientists had already made the genetic sequence of the coronavirus available to scientists around the world, and we actually received it in Oxford on the 11th of January. They'd released it a few days beforehand. And just to give a sense of how critically important it was that sequence was shared at that early stage, you can see that from the newspaper headline, which says this was the first death from a new virus, and the small print, which you might not be able to read from the back, says essentially, don't worry because there's no evidence that this virus spreads from person to person. So at that point, it was already possible for vaccine developers around the world to start thinking about how to make a coronavirus vaccine. And over the course of the next few weeks, it became apparent to all of us that this was a virus that was spreading person to person. We very quickly, by the next few weeks, by the beginning of February, knew that it particularly affected older adults. We had a good idea about transmission. As more data came in from China and then eventually from Italy, it became possible to start populating mathematical models to give us a good idea of what would happen if the virus was allowed to spread unchecked by non-pharmaceutical measures such as social distancing and lockdowns. And in fact, those were then the first tools used because we understood transmission, we understood that this was a respiratory virus and how it spread. So very different from the situation in 1347. Over the next 11 months from that point, there were 3 vaccines licensed by, well, about 11 months later, Pfizer, Moderna, and AstraZeneca. And those 3 vaccines, I think many of you will think maybe were developed too quickly. But of course that ignores the fact that there was 20 years of research on coronavirus vaccines that allowed the last bit of the effort, the clinical trials, to progress rapidly. And I'll just explain what I mean by that. But rather than thinking of it as 11 months of development, it really was 20 years and 11 months that got us to that point. If you think about the technologies that we use for the Oxford AstraZeneca vaccine, using a chimpanzee adenoviral vector, the first viral vector vaccines were developed over 40 years ago, and for hepatitis B. And there's been lots of work using them, many clinical trials using viral vector vaccines, and even several licensed viral vector vaccines now for Ebola, before the COVID-19 pandemic, when there are 4 viral vector vaccines which were used in different jurisdictions around the world. The RNA platform that Moderna and Pfizer used to great effect really has had very active research for more than a decade, and there were clinical trials of multiple products underway before the pandemic. And then relatively trivial for both viral vectors and for RNA vaccines to go from knowing the sequence of the virus to making a vaccine for them. And the reason why there's so much experience of coronaviruses is because over the course of the last 2 decades, we have had very serious threats from coronaviruses. About 20 years ago, SARS caused cases starting in the Far East with a mortality of around about 10 or 11%. So very worrying that this virus had emerged. Fortunately, it didn't spread very well. There wasn't much of an asymptomatic phase, and it could rapidly identify cases and isolate them, and that epidemic was halted by public health measures. But then just over a decade ago, Middle East respiratory syndrome, another coronavirus spread from camels, emerged in Saudi Arabia. And this virus, at least in the cases that were documented at the time, had a mortality of about 30%. And so the international community were really concerned that a new virus had emerged in humans that would kill 1/3 of individuals. Fortunately, again, this virus didn't have much of an asymptomatic phase and didn't seem to spread very well between people, so it could be halted in its tracks using public health measures. Important to say here that this virus hasn't gone away, that there were cases continuing over the decade up until the pandemic. And although, for reasons which are not entirely clear to me, during the pandemic the numbers of cases have reduced, this coronavirus is still out there, it's still in camels. It just hasn't yet mutated to be able to spread readily between people. And because of that severe threat that MERS posed, the Middle East respiratory syndrome virus, in Oxford we'd been working on vaccines for that virus over the 5 years previously, funded by the UK government initially. And my colleague Sarah Gilbert had already made the vaccine. It was in phase 1 trials completed in the UK and were also being run in Saudi Arabia. So when the pandemic arrived, there were already coronavirus vaccines like ours being tested in humans. And with these vaccines, all you need to know is the sequence of the key antigen, spike protein, from the new coronavirus, and you can just insert that into your vaccine platform, and you have a vaccine for the new pandemic virus. So for coronaviruses, we knew how to make a vaccine, and we're extremely well prepared for that relatively trivial step once you have the sequence of the new virus. And then, of course, we started to realize over the course of the month from January of 2020 that a pandemic was upon us. And these are some figures from an analysis at the end of 2021, whereas at that time there were somewhere around 5 or 6 million deaths documented by WHO from COVID-19. Of course, those are proven deaths where we knew that the cause of the death was coronavirus. But the actual deaths, when you model the likely missed cases, were already at that time thought to be around somewhere between 18 and 20 million. So the virus has had a huge global impact as a result of its spread around the world. And the challenge when we look at the media reporting, and in fact the many scientific commentators at the beginning of 2020, was a view that vaccines were unlikely to play a major role in curbing the pandemic. And that really came from the models for influenza. And you may recall that most influenza vaccines used in the world are made using eggs. And that manufacturing process takes at least 6 months. So if you think about unchecked waves of a pandemic virus, where you have to grow the vaccine virus in eggs, by the time you've actually made the vaccine at scale that you can deploy it, most people who are going to die will already have died if the pandemic is unchecked. And so many commentators said, well, we won't have a vaccine for at least a few years, and therefore we have to look at other measures to manage the pandemic. And so a lot of the early focus was on trying to reduce transmission so that the pressure on health systems was managed, but accepting that as we tried to make vaccines or new treatments, that we may very well end up with many individuals still dying or ending up in hospital. But at least if you could keep the system running by managing over time the number of those individuals going to hospital, and then the health system could cope. And it's not an unreasonable view to have taken. If you look at this, this is what typically happens with vaccine development. The research and development, like with coronavirus vaccines, can take decades. Some vaccines are much more difficult than others, but once you have worked out how to make the vaccine, you can then move into manufacturing. The problem with manufacturing is we have very limited capacity around the world, and often over the last 20 years when I've been starting trials of new vaccines, I finally got the funding to make the vaccine to start my phase 1 trial, and the next slot available in a manufacturing facility is still 18 months away. And then when you come to the next step and you've done your phase 1 trial and you want to move on to the next trial, you have to apply for a grant to do the next trial, and that takes a year before the funder decides whether or not to give you the money. And 5 years goes past incredibly quickly in development, not because of regulatory hurdles or because of major scientific obstruction to moving forwards. It is just the logistics that are a major problem, which is why for most vaccines that we currently deploy, the period for development is often 10 to 15 years. So I'll talk a little bit about the timelines of the development of the Oxford-AstraZeneca vaccine, but it really represents a similar story to the other vaccines which have been developed. And we were working very much in parallel with Pfizer and Moderna and Johnson & Johnson, all of whom had vaccines that were widely used in Europe and in other Western countries. And the first part was to make the vaccine. And as I mentioned, designing the vaccine is relatively trivial. It took a couple of days to design the vaccine because we already knew how to make coronavirus vaccines. But then there's a period of work that's done in animal studies to make sure that the construct is expressed and then induces immune responses. And finally, that the vaccine that we developed protects animals. So before even the first dose was given in humans, we already knew that the vaccine was 100% protective in our animal models, even in non-human primates. And the vaccines were then finally released to trial with all the regulatory checks done by the middle of April. And so the phase 1 trials could get underway on the 23rd of April of 2020. And those trials, we had an overwhelming response from the public. We opened a website to take bookings, and we couldn't cope with the number of bookings which were coming through to take part in the clinical trial. I wish it was like that with all vaccine development that I've been involved with. One of the other critical things for vaccine development is having the critical mass of people and capabilities. And I think the reason why Oxford University was the only university that was involved in putting together a vaccine all the way through to licensure in collaboration with AstraZeneca, was because we had many vaccine projects driven by passionate investigators who were working on a disease that they were interested in. So that when COVID-19 arrived, those individuals could come together and focus on what was needed to combat the COVID-19 pandemic. And so what we did is we put senior people in charge of the clinical team, in charge of manufacturing, in charge of the animal studies, but taking the staff from all of those other teams together to provide the critical mass. And with that, the first studies showed that the vaccine made very good immune responses in humans, and in fact 2 doses were better than 1. We'd originally been advised by the modelling community that the pandemic would be so rapidly spreading that there was no point in looking at a 2-dose schedule because we would only have time to give the first dose if we wanted to have an impact on the pandemic. And so we did look at 2 doses, but originally were planning a 1-dose schedule. Of course, what then happened was the non-pharmaceutical measures were far more effective than expected. The modelling hadn't predicted that populations would be so compliant with the order to stay at home and that spread would be halted in the way that it was here in the UK. So antibody and T-cell responses were made by the vaccine. But even 2 days after that very first dose was given in the clinical trials, it became apparent that we were in a very different world from normal. On the first dose was given on a Thursday. On Sunday morning, a fake news website said that the first volunteer in the trial had died. And that story was then projected all over the world, that the first volunteer had died. Now, in itself, because it was a fake news website, doesn't seem a big issue, but the way that was then reported meant that the friends and the family of the volunteer were getting the news that she had died. And fortunately, a number of the media outlets, including the BBC, very rapidly on a Sunday morning did a proof of life interview with her and were able to debunk the fake news website information that was being reported. Unfortunately, there isn't penetration of the truth to all corners of the world, and there are some countries which still think that this first volunteer didn't survive. So those first clinical trials were reported in July of 2020, and we then, at that point, continuing with the theme of the media, started to receive huge numbers of contacts from journalists, from the major media outlets, from every newspaper in every town in almost any country in the world, to a peak of 1,000 contacts per hour from global journalists wanting to know more about what was happening and reporting and misreporting what was going on. We moved on to phase 2 studies showing that the vaccine made good responses in older adults across all age ranges. Of course, there is a healthy elderly effect here in that those who, in the middle of the pandemic, could come to a trial centre are much healthier than individuals who are too frail to leave their homes. And the logistics of actually running the trials at homes was too difficult. So we do know now that although in healthy older adults, immune responses are pretty much the same as in young adults, that in the most frail, with any of the vaccines, we see lower immune responses and slightly lower protection. And as this work was going on, there was more fake news. We had lots of amplification of negative stories, both directly from malign actors, as well as by chatbots which were being deployed on a huge scale, particularly in Europe and the US, affecting not just our vaccine, but other vaccines as well. As we started the phase 1 clinical trials, it was already apparent that we needed for regulators to have data on how well the vaccine worked at scale. And so I started contacting colleagues all across the UK, in South Africa and Brazil, because we wanted our vaccine development to be representative of the different continents, the different ethnic groups around the world, as well as different social circumstances, and to know that for all of those different regions, that the vaccine would induce similar responses and similar levels of protection. And so overall, more than 25,000 individuals took part in the clinical trials in Brazil, South Africa, and the UK, with almost 2,000 researchers across those countries running the trials, collecting blood samples, shipping them to the laboratories where we were testing immune responses, and working with the local hospitals to work out whether those who were vaccinated or those in the control group were protected from coronavirus. To me, this, when you look across what happened with our vaccine, with Pfizer, Moderna, Novavax, many other vaccines in development, several 100,000 members of the public took part in these clinical trials, and probably the greatest visible global public effort for public health that has been in our generation. Quite a remarkable achievement. And not least when you think in each individual, the researchers were saying to them, we don't know whether this vaccine will work, we don't know if it's safe, can you come and take part in the trials? And people were willing to do that because of the huge impact that they could see that a successful vaccine could have for the pandemic. It took about 300 days from the starting off with the genetic code of the virus to actually having data to submit for licensure. And so that's the sort of timeline that is possible if you've done 20 years of homework beforehand to be ready to go to deploy the vaccine. I mentioned the importance of partnerships with our colleagues in other continents, the academic groups who we were working with, but we also made a partnership with AstraZeneca in May of 2020. And that was a very important partnership because universities are not very well set up for global manufacturing and distribution of vaccines, which AstraZeneca was extraordinarily successful in doing. The other thing which I think was critical with working with AstraZeneca was that they were prepared to come in with Oxford with a view that the vaccine should be not-for-profit. Now, that is an extraordinary decision from AstraZeneca to agree to that, and particularly now with hindsight looking at the huge profits that the other pharmaceutical companies have made. But the reason why it was so important is, as a university, we were not interested in the concept of profiteering in a pandemic. But it was also important because if you have profit as the cornerstone of vaccine development, that means you focus your sales on only high-income countries and anyone who will buy that vaccine, even if it's those individuals at very low risk from coronavirus. Whereas in a not-for-profit model, it makes no difference to the company whether they sell to an African country or a South Asian country or a high-income country. There is a very small amount of revenue that's possible in all of those regions. And so the incentive is not perverted to focusing just on where the market is rather than where the need is. And when you look at the global burden of severe disease and death, it's almost all in individuals over 65 in whichever country you're in. And when you exclude high-income countries, the population of over 65s is actually pretty small in the world. And yet the vast majority of those never received a vaccine dose because of this incentive for most developers to vaccinate first of all younger and younger adults and then even children in high-income countries before those at greatest risk elsewhere could be vaccinated. AstraZeneca also ran trials in the US, in Russia and in Japan, providing data for regulators elsewhere, and forged a partnership with Serum Institute of India, the world's largest vaccine producer, which meant that we had huge scale of manufacturing. Now that was perhaps the biggest manufacturing site, but what AstraZeneca did, which is very unusual, is supported vaccine manufacturing in 24 other locations around the world, so that to some extent supply chains, manufacturing capacity was protected in different regions of the world so that vaccine doses could still get to people where they were needed. That is the most enormous effort. Vaccines are very complex biological products, very hard to get even two sites producing the same vaccine in the same way. And so a huge, heavy lift to manage that to meet the regulatory standards globally. So after the vaccines were licensed, first here in the UK by our regulator, the MHRA, on the 30th of December, we then had doses deployed here in the UK in early January, and the European Medicines Agency a few weeks later approved the vaccine, which then led to the World Health Organization approval called pre-qualification, which meant that the vaccine could then be deployed through COVAX. And I'm particularly proud of that moment when the World Health Organization approval came through, because at that moment, almost all vaccine doses that were being produced were going to high-income countries. And in the first half of 2021, 90% of the doses that were delivered to low-income countries through COVAX came from our partnership with AstraZeneca and Serum Institute of India. And of course, the critical thing with vaccines is you have to vaccinate people before they're dead. Otherwise, they're of no use. And most of the ability, particularly in low-income countries, to protect individuals from coronavirus was lost by later in 2021 because the virus had spread through all of those crowded communities. And so that early deployment of vaccine is critical. And we know that here as well, that vaccinating people early in the pandemic, early rollout, saves far more lives than a slow rollout. And here you can see just some of the many headlines about doses being delivered all over the world. There were many headwinds that vaccines have faced. There was a very poor understanding of variants as they first started to emerge and how they were escaping from natural and vaccine immunity. We have a very clear understanding now about these. I don't have time to speak in detail about that at the moment. But perhaps the biggest headwind for the Oxford vaccine was the discovery of a very rare side effect, thrombosis and thrombocytopenia syndrome. And that had huge implications, particularly in Europe, for use of the vaccine. But here in the UK, the risk-benefit calculation was very carefully, I think, communicated to the public at a time when we were just about to experience the Delta wave, so that whilst there was limited supply of vaccine, the vaccines were very carefully deployed to those individuals who were at the greatest risk to make sure that the benefits of vaccination were managed. And now when you look at the impact of vaccination, you just realize how astonishing and powerful vaccines are and how persistent that protection is. We tend to look at cases rather than the severe disease. And cases, as we know, are extremely difficult to prevent with vaccines against respiratory viruses, because most viruses that are very successful in humans are able to adapt to the immunity that we have in our upper airway. But what we're really interested in, and what we had a pandemic of, wasn't of colds. It was a pandemic of severe disease, hospitalization, and death. And what you see on this plot here is, on the left is the first wave of disease with the original virus that first arrived on the shores here, and a very large wave of deaths. And then towards the end of 2020, we had a very major wave that started to kick off in September and was then accelerated by the emergence of the Alpha variant, which at the time was known as the Kent variant because it was first discovered in the southeast of England. And then the vaccine rollout happened. And it was, most of it, particularly in those at risk of dying, was completed over the first 5 or 6 months of 2021. And what you see here is that the pandemic of deaths ended as soon as that rollout had happened. So although we've had Delta, we've had multiple Omicron variants that have arrived on our shores since then, the pandemic really ended, if you describe it as a pandemic of deaths, in the middle of 2021. Now, you wouldn't believe that from what you read in the media, and in fact from much of the scientific commentary that seems to imply that the pandemic has been continuing. But if you work in the hospitals, from the point of Delta, we saw a complete change in the spectrum of patients who were coming in and the pressures on the system, because variants although they emerge to allow the virus to survive and to transmit, they're not emerging in a way that results in severe disease or death. And in fact, it's almost unthinkable that that could happen with mutations in this virus, because the virus that we're dealing with is now one that is infecting us in the face of huge human immunity, vaccine-induced immunity against a very large protein, but a virus that's only mutating a few bases within that protein each time. And now we've got hybrid immunity from infection, which gives us very strong protection. So if we now look during that first year of the rollout of vaccines globally, at this analysis from Imperial College, it looks as if somewhere approaching 20 million deaths were averted just in 2021 as vaccines were rolled out. And the analytical firm Affinity has tried to break that down across the different vaccines which were developed. And what you see here is the major contributions from Pfizer and AstraZeneca. AstraZeneca, I'm pleased to say, just winning in this analysis, which I think really represents the importance of getting doses, large numbers of doses, to those who are at greatest risk of dying as early as possible to have an impact. So all of the vaccines are needed to have the huge global impact that there was. And these are the vaccines in 2021 which were deployed in a way that could have the biggest impact because they were before the virus had spread to all populations. We are quite unclear on what the level of protection is that's required to prevent severe disease. It's probably much less than is needed to prevent infection. We do know, at least with the original virus, how much antibody you need to prevent infection. But we don't know with the new variants as they emerge what exactly is needed in terms of levels of antibody to prevent infection. And I just want to briefly go through some interesting data that perhaps explains the issue with variants to you. So these are the lungs of hamsters that have been exposed to coronavirus. This one here is a hamster that's been exposed to the Kent variant, and this one here is a hamster exposed to the Beta variant, which you may recall emerged in South Africa. And in both cases, these are very abnormal lungs. You can see virus in the tissues and an inflammatory infiltrate in the lungs. Now, if you vaccinate with the vaccines which were originally developed, a separate group of mice, you can make very good immune responses against the alpha variant, but you find almost no response measured in neutralizing antibody against the beta variant because the virus has mutated to escape from neutralizing antibodies, the antibodies that we think are important for protection against infection. So with the alpha variant, you'd expect with these high levels of antibody for the animal to be completely protected, and that is the case. These are normal lungs, there's no virus in the lungs, there's no inflammatory infiltrate. So vaccination works in preventing disease caused by the alpha variant. But with the beta variant, you might expect that the hamsters would not be protected, but actually they also, vaccinated hamsters with the original vaccines, have completely normal lungs because the vaccine is still able to induce immune responses which protect the lower airway. No virus in the lung, completely normal tissues. So that is like us. This is human populations today who've been vaccinated. We have very good levels of protection in the lower respiratory tract. But interestingly, if you look in the upper respiratory tract, you can find virus in the beta variant exposed hamsters. You find very little virus in the alpha exposed hamsters. So the upper airway is a different immunological world from the lower airway in terms of what's needed for protection. And we really don't know today what sort of antibodies or T cells are required to prevent severe disease, which of course is the critical thing for preventing pandemics. That's a huge gap in our understanding because we don't actually know what we need to do to prevent this pandemic. There's a lot of real-world data which helps us understand the performance of vaccines. I'm going to skip over this because it makes a similar point. But there's a lot of real-world data that tells us about performance of vaccines. And I think the main message here is that if you look at severe disease, almost any vaccine provides high levels of protection compared with nothing. And so there were lots of criticisms of, for example, the Chinese vaccines, which are inactivated vaccines, rather like the polio vaccines that we use today. But those vaccines were deployed extremely early. They particularly got their doses to low and middle income countries. And the protection, here you can see with one of them, CoronaVac, was extremely good, not so different from the RNA vaccines or the viral vector vaccines that we're more familiar with here in preventing severe disease. They weren't so good at preventing infection, but the criticism of them, I think, is unwarranted in that they saved many lives as a result of their use. And even if you now take one of those inactivated Chinese vaccines and you boost it, you get very high levels of protection against death and severe disease that you can see here on this plot. So the vaccines that we have are remarkable tools in preventing severe infection. So I started by talking about the pandemic of 1348 here, but it would be wrong to think that the pandemic caused by Yersinia pestis, the plague, is the only pandemic that's been threatening human populations since then. We've had many others listed on this slide. The last cholera pandemic finishing only in the 1960s. The last plague pandemic finished in the 1960s. We've had 6 influenza pandemics in the last 150 years, smallpox, HIV, and so on. Many of those had much higher mortality than we suffered with COVID-19, and in many ways you might consider COVID-19 was a relatively mild pandemic as pandemics go. It didn't feel like that, and of course there were many families very severely affected by it. But compared with what might have happened, it was a relatively mild pandemic. And I think there is some slightly more worrying ways of looking at this. First is that there were almost 350 vaccines that have been in development, but the World Health Organization has only been able to approve 8. So there's a very large number of failures because it's actually quite difficult to make vaccines. There are lots of challenges along the way, not just the scientific ones, which, as I've said, are in some ways relatively trivial, but actually getting from that scientific idea through to producing a vaccine and making it at scale is very difficult. And we are continually exposed to microbial challenges around the world that threaten our civilization. And of course, this is avian flu, which there is currently an ongoing avian flu pandemic currently affecting birds that only needs a few mutations to become a virus that spreads in humans. It's not difficult to imagine that vaccines have a huge impact from the huge amount of data we have from previous vaccine programs. But I want to just end by talking briefly about where that leaves us. Well, if you go back to 1348 and try and understand why did we have a pandemic of plague? Well, it's pretty clear now from experts in this area that climate change at the time in the mid-14th century changed the behavior of rodents, which meant that their fleas spread to new populations. And that impact of climate change fueled the start of the pandemic of plague, killing half of the population of Europe. And of course, climate change today is already having those impacts. We're seeing insect-borne pathogens spreading to new territories that we haven't seen them in living memory. And that, including here in Europe. So I think that already is quite worrying that we're at a point where new pathogens are arriving in populations that have not seen them before. The population of the world currently is at about 8 billion. Over the next 30 years, we'll be at 10 or 11 billion. So crowding on the planet will really have a big fueling potential for future pandemics. And of course, those people will be encroaching more and more on animal habitats. We've seen outbreaks of plague in the US, in China, in Russia, in Africa, over the course of the last few years. So even something, an ancient disease like that, can cause small outbreaks. And of course, we travel a lot, including to conferences like this one, which would aid a spread. There are many pandemic threats, and there are lists that governments have, that non-governmental organizations have. This is the list from the Coalition of Epidemic Preparedness, and if you look at their full list, they've got 100 different viruses that could threaten human populations. Here in the UK, our government has a priority list of pathogens, and there's a funding call out at the moment for vaccine development against this priority list. For many of these, we are at an earlier stage than we were with coronaviruses, but there is some progress in trying to understand how we might make vaccines for these. We also are now in a unique position in having a much greater understanding of the performance of all the different vaccine platform technologies, proteins, viral vectors, RNA, inactivated vaccines, protein polysaccharide conjugates, which will be relevant to the different types of pathogen. And we have to maintain our abilities to generate vaccines across these different platforms, because for different pathogens, viral vectors or RNA vaccines may not be the appropriate platform. When we look at coronaviruses, we know now that if you take the spike protein from coronaviruses, we've got examples across multiple coronaviruses that you can make a vaccine. We don't know that for all viral families. If a new virus emerges from one of those other families I listed, we wouldn't be sure at this moment that taking the glycoprotein from that virus would then induce a protective immune response. And what about a virus that we've never seen before that jumps from an animal species that we haven't worked on? And what about bacteria, which, like plague, are much more complicated for making vaccines? And then how will we know they work? If the disease isn't here at the moment, we can't test efficacy. So we end up having to use animal studies to predict how we might move forwards with those new products. And then finally, how will they be commercialized? There's no market for vaccines to prevent a disease that isn't here at the moment. And that really produces huge challenges for industry to even begin to take on developing vaccines for new pandemics. And that leaves it to academics and governments to do those first bits of research, but are not going to get us to a point where we're ready to go rapidly with the 100-day mission, which is currently the view of the G7, the richest governments in the world, that we need next time to be able to deploy vaccines at scale within 100 days of a pandemic being declared. That's a really tall order with the lack of information that we have against many of these viruses at the moment. In my group, there's a large number of investigators working on some of these pathogens. And really, I wanted to end just by saying, we've had this huge impact with COVID-19 vaccines. We know what we need to do to prepare for the next pandemic. And we really have to focus on how do you maintain those capabilities for response in peacetime, in manufacturing facilities, in academia and in industry, so that we are better prepared to go. I really do think science has got this, but the problem at the moment is that huge knowledge we have is not sufficiently invested in to make sure that we're ready if the next pandemic comes tomorrow. Thank you.
So thank you very much, Andrew, for a really excellent talk and taking us through the development of the vaccines. I'm sure that's generated lots of questions. We certainly have time for a few. Please, Jeff.
Thanks, Andrew. Jeff Adamson, Oxford. That was a superb resume. These vaccines are clearly hugely safe. There are very rare adverse effects. And it would be good if those effects, rare though they are, could in some way be predicted. Is there anything going on that helps us understand the mechanisms and how to get on top of those rare effects?
That's a great question. I mean, so the very rare side effect I guess you're talking about with viral vectors is the thrombosis thrombocytopenia syndrome. And there's been quite a lot of work trying to understand that condition. We actually know quite a lot now about the downstream mechanisms of how that is generated, almost certainly through the generation of antiplatelet factor 4 antibodies. What is not really understood is what it is that you would change about viral vectors, and it seems to be true with the adenovirus viral vector from Johnson & Johnson as well as the AstraZeneca viral vector. We don't know how to change the viral vector to stop that occurring. And even if we had laboratory evidence that suggested changes, and there are some ideas out there, how do you then get to the point of demonstrating to the regulator that the changes you've made have now prevented that from happening? Because we don't have an animal model that reproducibly produces that side effect. So I think there's a major challenge from understanding some of the downstream mechanisms to getting to a point where we could actually make a change. Of course, then it would be a new product. You'd have to go through the whole licensing process again. If there was another pandemic tomorrow, the world would need RNA vaccines, viral vector vaccines, inactivated vaccines. We're not going to have a choice apart from to use them again, because that is the current status of capacity to protect human populations.
Michael.
So I've got two questions. First is that I've got a grant for a big EU problem, which is whether long COVID causes neurodegeneration, because we've been able to show the same enzymes involved in ALS and Parkinson's are crucial for viral entry and exit, and there's a real risk for that. So my first question is, and we're having a meeting in 10 days, so your answer is very important. Do the vaccination really change long COVID, and will there therefore be links in terms of protection for any neurodegeneration which may come about subsequently?
Well, that's a lovely question. I wish I knew the answer to it. So in terms of, I'm not sure whether you meant treating long COVID.
Sorry, yeah.
So as far as treatment is concerned, as you're aware, there's lots of anecdotal reports of people getting better after being vaccinated. I'm not sure whether that there is robust enough evidence to address that question at this moment. It does seem to me, though, that if you're vaccinated and it reduces your risk of COVID, then that may be quite good at preventing long COVID. Of course, it does seem to be less common amongst people who are already immune than we saw originally, but when everyone was naive. So I think in that respect, vaccination is good for long COVID, but as a treatment, I can't really answer.
And the second question is that the rump of deaths, if it's oxygen-dependent disease to a certain extent when it's severe, people in nursing homes have got a VO2 max of about 13, so they've got no oxygen-dependent reserve. So are we going to see a long-term reduction in long-term life expectancy in humans from residual COVID, which may or may not be treated by vaccination?
Well, I think that's a very interesting question as well. Very clearly, in the naive individuals who were not vaccinated, who got severe COVID, they had a very significant low respiratory illness. And I mean, some of those individuals are being followed, and there are changes in lung performance in those individuals. In the milder cases, I think it's much less clear. But in those who have been vaccinated, we really don't see significant low respiratory disease. It's mostly a miserable upper respiratory illness. And even today, where particularly in older adults, recommendations for boosting to try to give additional protection against variants, that's not about preventing lower respiratory coronavirus replication. That's about stopping someone with heart failure from deteriorating because they've got an upper respiratory infection. So I think, obviously a worry about people who are severely affected early, just as with any severe respiratory illness, but I'm not sure in a vaccinated population this is as big an issue as one might imagine from the number of cases.
Great.
Thank you. Number one, here.
Sajak here from Southampton. Thank you very much. That was tour de force. You showed in the slides that the different platforms may be suitable for different infective agents as we develop them. In retrospect, regarding the COVID-19 platforms, you said that all prevented deaths in serious disease, so one can take that you didn't think the platform matter in the sense of preventing deaths in serious disease, but there were differences in in mild disease, possibly long COVID. In retrospect, do you think we have data to say, did either of the platform has the marginal advantages, or both of them had pros and cons, and really one cannot select a better one?
It's a very good question. I mean, I think that, you know, when you get into the minutiae of differences between the platforms, viral vectors are better for T-cell responses than RNA. RNA gives higher antibody responses, but those very high responses can't be sustained by the immune system, so they tend to drop quite rapidly. It's very difficult to say whether there is one platform you would advise over another. And the differences in the ability to block infection is sort of something that's very relevant today, because today we're, like with flu, we're trying to get a short-term rise in antibody for a season. With flu, we know when that season should be, and so you can vaccinate in the autumn and protect those at risk for the next few months during the season. For coronavirus at the moment, I can't tell you when the next wave will come, because it hasn't become a seasonal pattern. So we've got an additional challenge with coronaviruses about deploying vaccine in a logical way, and also we don't have any way of predicting what the variant will be, whereas at least with flu, we've got some ways of predicting what might circulate.
Thank you very much.
Gilberto Danucci.
Okay. Thank you for excellent talk. My question is, you said that we have been prepared for this SARS-CoV pandemic because we had some data from 20 years. We have HIV going for 40 years, and apparently not so successful in terms of vaccination. Can you comment on that?
Well, I mean, HIV is very different. It's a much bigger challenge than coronavirus is because of its very high mutation rate, both within an individual host as well as between people. And that's the difficulty. It's, you know, if you could stabilize HIV so it didn't keep changing, you could make a vaccine that prevented infection. But with coronaviruses, to prevent severe disease, which is a low respiratory infection, it seems much easier because that immunity doesn't seem to be a problem for the escape mutants, which allow upper respiratory infection. So yeah, it's not for want of money in trying with HIV. It's a much more difficult target.
So a final brief question from Danny Hoyer.
It's not a question, it's a recommendation for the 100-day plan. Melbourne had 270 cumulative days of lockdown, and by that time Australia and New Zealand, for that matter, were completely isolated. And these people thought they were completely protected as well. It took them another 4 months to register the vaccines in the countries when everything was already there. I mean, there was no point in delaying the thing. Hence, vaccination was delayed. And the people felt this, since they were isolated, they were protected. And by the time the borders opened again, then you could see the rise in infection and the death that came much later than in any other place. So there's this misperception that by isolating yourself, you're going to be protected. And Western Australia is the worst of all examples, because these guys were even worse, and yet it came. So bottom line is, once you have the vaccines, use them as soon as feasible, and just don't think that you can isolate yourself and just be safe.
Well, perhaps just a comment on that the zero COVID policy from a public health perspective is quite a good one to buy time whilst you're waiting for vaccine development. But as you say, once you've got vaccines, you need to vaccinate. The problem also is that, and I think this is the dilemma for politicians, is that all of those of us in public health are very keen on zero COVID, no transmission, and then the vaccine's available and you can vaccinate people. But they've got other issues to worry about. They've got political issues, they've got the economy, and we now know the huge impact, and in Western Australia particularly, that has on children and on the economy if you have very prolonged periods. So it is a very difficult balance. From a public health perspective, I always think it's much easier. You just want to stop transmission until you've got a vaccine. But the real world is more complicated for the politicians to navigate than that is.
Good. Well, thank you very much. It's time just to sum up this session. So thank you very much again, Andrew, for all that you've done to help protect so many people and continue the good work you're doing with other viruses, and as you say, bacteria, and I think also fungi. We've got to start to worry about them as well. So thank you very much in your busy schedule for finding the time to come up. Thank you to the audience for being here and the questions, and enjoy the rest of your day. Thank you.
Thank you very much.